I'm Paolo Casale from Helmholtz Munich, and today I'm going to present some of the work that we do at the interface of human genetics and machine learning. I'm going to start with something we all agree on. Genetics is a special layer. It's a powerful tool that we can use to identify biology that is upstream disease. And this is because we inherit our DNA at conception. It is constant throughout our lifespan, and also mostly across all the cells in our body, and we can measure it very precisely. And we've known this for quite some time. It's now 20 years that we do genome-wide association studies, where we collect phenotype data and genetic variation data from a large set of individuals, nowadays in the hundreds of thousands of samples, even millions. And then we apply a simple regression test between every single genetic variant in isolation and the phenotype of interest to find associations. And a typical representation of the output of this analysis is a Manhattan plot. In this plot, each circle is a variant. On the x-axis, you have the position of the variant. On the y-axis, you have the strength of the association with the phenotype that's typically measured in -10 p-value from the association test. And so this Manhattan is from a relatively recent GWAS of Alzheimer's in 2019. And you can see that this type of analysis find different regions in the genome associated with disease risk. You also see that the closest gene to the top variant. But the challenge here is really how to go from these lists of genes and variants to actual disease biology. Because even if we now know which regions are associated, we still don't know what's the right tissue, what's the cell state, what's the cell type, and what's the functional mechanism throughout which this variant ultimately affect disease. And so what we do in the lab, we try to bridge genetic signals to disease mechanisms by leveraging high-content phenotypic data and machine learning. And we do this across cell traits, organ-level traits, and clinical readouts, working at all different scales and with different modalities. Today, I'm going to tell you about the work that we do on histology data and also a bit on retinal images. And that's in the first part of the talk. And then in the second part of the talk, I can tell you a little bit about work that we did in genetic causal inference for risk prediction, where we try to connect these intermediate phenotypes to disease. So that's the agenda for today. So let me start telling you about the modality we work with. So here it's H&E stained all-slide images. These are microscopy images of tissue sections. They are commonly used in the clinic to diagnose cancer and also liver inflammation, chronic inflammation, steatohepatitis. And they do showcase a lot of cellular and tissue level phenotypes. And so these are, they contain millions of pixels, but if you zoom in the images and you zoom again, then you can start seeing some interesting disease-related phenotypes. Like here on the left, we see hepatic death. We see hepatocyte dying. On the right, we see the presence of connective tissue in the liver. And these are two phenotypes associated with chronic liver inflammation. And so despite this richness, though, at the moment, there is no method that enables to perform a systematic genetic analysis of these traits. And so that's the gap that we wanted to fill, and we propose an approach that we call HistoGWAS. So here we build a semantic autoencoder to project these images of patches from these histological images to a latent space. And for this, we use a pre-trained foundation model, as we will see soon. And then we learn how to invert this foundation model and decode these latent representations back to the image space. And then in this latent space, and we call embedding space, we can do a genome-wide association study. So it is very similar to the type of analysis that I was showing you before. But now we test for association between every single variant and the whole embedding. And then finally, if we have genome-wide significant hits, we can use the decoder for interpreting the genetic effects on the image. And we do that because once we know the direction of the genetic effect, we can do linear interpolation in the latent space and use the decoder to visualize these interpolations in the high-dimensional space. And this is a methodology that is very common in the machine learning community. You may have seen these, people interpolating between people's faces. And so we did this analysis in the GTEx dataset. This contains expression, genetics, and histology from 54 tissues in 1,000 individuals. And we specifically analyzed the 11 tissues with the highest sample size in the cohort. So go a bit more into details on these three steps. So I want to start very briefly on standard autoencoders. Right? These contain an encoder and decoder that you train jointly, directly minimizing reconstruction loss. So when you do that, of course, your latent representation wants to preserve all the information in the image for accurate reconstruction. We wanted to do something slightly different. We want to only preserve information that is interesting to us. We'll call it, we want to present a semantic information or content in the image. And to do that, we thought maybe we should use a pre-trained encoder, maybe enough, especially with all these different foundation models. And then we actually spend computer power to invert this pre-trained encoder by training a generative decoder. And so just one example on how this encoder may be pre-trained through contrastive learning. I think that's a very good example for this application. So this is the SimClear training strategy. And so if these are two images of patches, histology patches, you can consider two different augmentations of the same image that do not change content, right? So the rotations, flips, color jittering do not change the content of the image. And then you train an encoder such that embeddings of different augmentations of the same images are similar, while embeddings of different images are different. And if you do that, your encoder learns to extract embeddings that are invariant to these transformations. And so they only preserve what we wish to define content here. And then once we have that, we then train a decoder that inverts this process. So starting from a real patch image, we can encode to get an embedding using the encoder that I was telling you before. Then we can pass this together with the Gaussian noise in a generative decoder to produce a synthetic image. And we train these against a discriminator. These are two players that compete against each other, where the discriminator needs to figure out whether an image is real or synthetic, conditional on the embedding, while the generator needs to make good images conditional on the embedding. And so the fact that these are trained conditionally lets the training them against each other makes the generative decoder learn how to invert the encoding process. So we train these across 20 million patches across 11 tissues. So this is how you put it all together. This is how it looks like, right? So you have your real data. You can use the contrastive encoder to get these semantic content representations. And then together with the noise, you can pass this to the generative decoder from the GAN, and we get this semantic reconstruction that they preserve content, but they're different in the realization. And you can actually change the noise and sample multiple realizations of the same context, which sort of gives you this nice decomposition of content. These are different rows and realization of the contents, where we only change the noise, right? And we get this way of decomposing the two concepts. And here, content is really defined by what the encoder extracts, right? So it depends on how you train the encoder, what you call content. So yeah, now how do we select the encoder? So we evaluated different foundation models, histology foundation models. We picked the one that best predicted expression. So in GTEx, we have matched histology and expression, so we can do that. Yeah, so here I'm showing auto sample expression prediction. This is a QQ plot. Each point is a gene. We train different models. This, this does not include the models that came out in the last year, which is quite a lot. In this comparison, the contrastive learning procedure appeared to be performing the best. Specifically, we picked red CCL, which is trained contrastively. Okay, now we have this procedure, right, of encoding, decoding. You can back and forth between these two spaces, but how do we evaluate the decoder? So we came up with this strategy where we can, of course, use the pre-trained encoder to go to the embedding space, and then we use the decoder and then the encoder again to get embeddings of the reconstructions. And now you can see how well these embeddings of reconstruction can actually predict expression versus the embeddings of the observed data. And we also compare versus embeddings of reconstructions from a standard autoencoder, right? It just minimizes reconstruction loss. And as you can see, here we preserve lots of signal when we use our strategy, while a standard autoencoder loses a lot of information present in gene expression. Now I go on to the second part here, how we did the genetic analysis. And one of the first challenge that you have, right? These genetic association studies happen across individuals, but our representations are at the patch level. So we have a representation for small patches of the full image. So how do we pull these patch embeddings into individual level embeddings so that we can perform this association test? So we use a very simple approach for doing that. Of course, I mean, the simplest thing you could do, you could just average, right, or the patch level embeddings. But yeah, we did something slightly more complex, but not that complex, where we assume that the genetic effects are local on this manifold. And so we did an unsupervised analysis and identified 68 morphological clusters across the 11 tissues that we analyzed. And then we take averages for each of these clusters, right? So we average within individual and within the same cluster. And then we perform a GWAS for each of these 68 morphological clusters. Yeah, we validated this a bit with a pathologist, also correlated with the expression, and showed that different expression parents associated with them. Go too much into this. How we did the genome-wide association study. So here we used a generalized linear mixed model with a binomial likelihood where the genotype is the outcome and the logit of the rate is modeled as an additive effect from covariates, things we want to account for. And then we have a random effect from these cluster signature embeddings. And within this framework, we can do this test very quickly because we can perform a variance component score test. And I just going to briefly make the reference here. This is similar to sequence kernel association test, but inverting genotype and phenotype for who knows this type of models. So these are the results. We get 4 genome-wide significant hits at a family-wise error rate 20% across 11 tissues. I'm going to tell you a little bit only about the top hit here, thyroid. So this is here now on the right, we see a log zoom on this hit. So it's about 1 megabase around the lead variant on chromosome 9. This hit was in the thyroid tissue. And when we look at the top variant is an intron variant of the PTC SC2 gene, the papillary thyroid cancer susceptibility candidate 2 gene. And this variant actually colocalizes with hypothyroidism in independent GWAS in FinGen. And we also then associated this variant with gene expression in the same tissue, and we find a module on 128 genes also regulated by these variants with lots of convergent biology. But I think that the novel thing is that now we can use the decoder to visualize what the variant does in the thyroid tissue. And so we showed these to 2 different pathologies together with the other hits, of course. And here we find that the G allele is associated with enlarged follicles and increased colloid. And this is likely a compensatory mechanism due to reduced thyroid function that happens during hypothyroidism. So it's one example in which you can see, you can study the effect of a variant in tissue, and you have a clear link with disease. So these are how the 4 hits look like, and you can use these also to make nice videos. Down there, this is the description that the pathology gave to these processes that you could see in the images. Great, so this is a preprint, hopefully, revision now, hopefully coming out soon. Now we're extending this work to study germline effects on breast cancer morphology in the Cancer Genome Atlas. And so we're doing, we're applying HistoGWAS pipeline to the histology images of cancer. Here, the hypothesis is that if you have a variant affecting morphology, it's an interesting variant potentially to also study effects on survival and prognosis more broadly. So that's the assumption that's why we're interested in this in the first place. But in this study, we also look at open AI embeddings of pathology reports from the same images, and then we do a GWAS of open AI embeddings, colocalize among these 2 modalities, and then prioritize loci that are multimodal with a series of downstream annotation analysis. We're also analyzing embeddings from a recent foundation model of the retina. It's a red found. So the original model, red found, is not our work. This was published in Nature 2 years ago now. And what the authors did, they trained this masked autoencoder on almost 1 million fundus images from a hospital in London, and then showed that with little fine-tuning, you can predict ocular disease and other systemic disease across different data sets quite robustly. And so now we are looking at the embeddings from this model for genetic association studies. So specifically, we fine-tuned the model a bit in the UK Biobank on the 50,000 individuals that have retinal images. And we fine-tuned that to predict age, because we want to prioritize processes associated with age under the assumption that this will better capture disease processes, at least for age-related diseases. Then we do multivariate GWAS on these representations. But in this work, we spend a bit more time understanding comparing different encodings and embeddings. So we compare the model where we do age tuning versus the model where you would not tune versus, say, just ImageNet pre-training. We also extended the evaluation, of course, to understand how much overlap from these GWASs we get with disease variants, looking at overlap with findings from the FinGen study, and also collaborating with a German mouse clinic to align what our generative model predicts with mouse knockout data and see whether we find consistencies. Yeah, so this concludes the first part of my talk. So I told you about how recent AI methodologies for representation learning and generating medical images can help perform genetic analysis of medical images and also interpret their findings. I showed an application to histology where we identified 4 genome-wide loci and spent a bit of time on one example of these. And then I didn't show you that, but we did a power simulation where we showed that increasing sample sizes, we see a great boost in power. So this shows the potential of this analysis once this data reach the scale of the thousands of people, also to help elucidate links between genetics and disease. So we know that these models, they give us powerful representations. And the next question that we asked can we do genetic causal inference on these representations, right? Can we use methods like Mendelian randomization to assess whether a specific direction in this representation has evidence of causality for a disease? And if that happens, right, we would find tissue processes that are upstream disease. So that brings me to the second part of the talk, where we'll talk a bit about genetic causal inference, giving a brief refresher on Mendelian randomization first, before going into our work. So say we see an association between C-reactive protein, a systolic blood pressure. Can we infer causality from these associations? And the answer is generally no, we can't, because there's confounders that affect both traits. This can be lifestyle, can be medication, it can be BMI. And so the gold standard for causal inference is really randomized controlled trials. And in this case, you take your samples, you randomly allocate your samples in two groups, controlled, they get the placebo, and a treatment group. They get a drug that lowers C-reactive protein, and you can do a statistical test to assess if there are differences in the disease outcome between these two groups. And if you see this difference because of the randomization that breaks the correlation with the confounders, if you see a statistical difference, then you have evidence of causality in this case of C-reactive protein, which is the trait that is targeted. And very similarly, we can do the same thing with the Mendelian randomization. Here, we use random segregation of alleles instead of the random allocation to groups. And so in the two groups here are people with a wild-type allele and people with the variant that actually lowers CRP. And then in a similar fashion, a statistical test between these two groups in disease outcome tells us about causality of CRP. So this is the assumed causal graph for causal effect estimation. Very briefly about the nomenclature. So genetic variant, I will refer to this as instrumental variable or instrument. Here, C-reactive protein is the exposure, while SBP is the outcome. And very briefly on the assumptions of MR. Where you see an arrow, there is an assumption, but also where you do not see an arrow, there is an assumption. The genotype must be robustly associated with the exposure. This association needs to generalize across different conditions. It needs to be independent of confounders, and there must be no horizontal pleiotropy, meaning that the genetic must affect the outcome Y only through the exposure. There must be no alternative pathway between the two. Okay, so given these assumptions, how do we estimate the causal effects? So we can write the structural equations from the exposure and the outcome. And then if we substitute the exposure X into the outcome equation, and we do a bit of simplification, we end up with this formula here where I introduce beta Y as beta X times alpha. And now if the assumptions of Mendelian randomization are met, you can estimate beta Y by regressing G on Y and beta X by regressing G on X. And then your an estimate of the causal effect is the ratio between these two estimates. But if you do a GWAS of a risk factor, and then you use clumping to identify independent variants from that GWAS, you end up typically with hundreds of genetic variants, right? So the typical settings that you have hundreds of independent instruments that you could use here. So can we use them for MR? So indicating with beta E, the estimated genetic effects on E, and beta O, the estimated genetic effects on O. Of course, you could compute this ratio for each variant, but more robustly, you can just regress the genetic effects on disease versus the genetic effects on the risk factor. And this is called inverse variance weighted regression. The reason is that you weight, this is weighted by the error on the outcome genetic effects, and that's the probabilistic model that underlies this regression. Great, but now if we do have multiple observed traits, X, or we have an embedding X, and we write our risk factor as a function of X, can we learn this function within this causal inference framework? Can we learn this function by directly optimizing the inverse variance weighted regression? And this leads us to the last part of the talk. So this is a primer, predictive modeling within the Mendelian randomization framework. And so what we did here, so we showed that if you have a healthy population cohort, like the UK Biobank, you typically have access to large variety of risk factors together with genetic data. And you also have external disease GWAS summary statistics for a disease you care about. You can actually train a disease risk predictor such that the genetic effects on this risk predictor fit the effect on the outcome in an inverse variance weighted regression setting. And so you can train the predictor using the summary stats from the GWAS of the outcome to actually train a predictor, and then you can use it. You don't need genetics when you use it in other cohorts for making risk predictions. Okay, so I mean nowadays it's sometimes it's simpler to define models just showing how you do a forward pass from the data to the loss, because then you just, if you specify what's your parameters, then you have autograd, and you can use Adam. So it's that's typically is enough to give a good intuition how the model works. So these are our data. We have k risk factors. Of course, we can do GWAS of them plus clumping, and we identify S instruments, genetic variants, and then we have a C covariates that we want to account for. And so of course, we can compute the risk predictor by applying a differentiable function on the risk factor. And then we can compute the genetic effects on this aggregate risk factor. This is just a closed form maximum likelihood estimator doing the SNP-wise regression on E, accounting for the covariates. And then we have the inverse variance weighted regression negative log likelihood, where we regress the effects on the aggregate exposure versus the effects on the outcome. And then we can minimize this loss over phi, the parameters of the function, the directional effect alpha, and sigma squared. So as a differentiable function, we use a relatively simple function. So we take a linear combination of transformation of single factors. We take specifically an ALU. And this is a nice function because it lets us model this phenomenon in risk predictions where a risk trait does not contribute to risk up to a certain threshold is met, and then the contribution escalates. And since ultimately when we do this regression, we are fitting the genetic effects on the outcome at the instruments. And typically, we observe hundreds, maybe max thousands of these. So the sample size is not very high. So in order to make a robust regression, we need to use a Bayesian formulation. I will not spend too much time on it, but we put a prior over the sum of the parameters, and then we optimize the elbow through black box variational inference. Okay, so not just flashing simulations. I think in simulations it works. Great. Now I'm going to drive you through the three applications that we consider in real data. So first, we aim to predict 5-year type 2 diabetes risk in the UK Biobank. So we built the cohort focusing on individuals without type 2 diabetes up to the initial assessment. And then as risk factors, we consider measurements at the initial assessment. They're here. And then of course, we have also, in the case of the UK Biobank, we also have the longitudinal labels. So we know who's going to develop diabetes in the next 5 years. And One thing that's important to note here is that we do not use the longitudinal, the follow-up data, the follow-up labels are never used for training. We only use the genetics, the genetic effects on the disease for training, and we use the disease labels only for evaluating the model. Yeah, we take summary stats, the type 2 diabetes GWAS summary stats. These are training data, and we train the model on 80% of the data, and then evaluated on the remaining 20% using the longitudinal labels. And it's actually quite nice to see that within a genetic causal inference framework, you can learn quite good risk predictors. We compared our model with a simpler version where we only model a linear function. So it seems like this non-linearity is helpful. And then also with the simpler alternatives that are based on classic Mendelian randomization framework. And of course, in this setting, we do not beat longitudinal prediction. So if we actually use the longitudinal data to predict, and we evaluate the model out of sample, this does not, this performs better than the methods that are built on causal inference, expectedly. Then we also looked at what the model actually learned when zooming in. And so here we see that, for example, A1C and glucose, they show significant increases when the trait meets clinical thresholds that are typically defined as risk thresholds in the clinic. That was also nice to see when inspecting the model. Yeah, then as a secondary application, we looked at the predictions of 5-year Alzheimer's disease risk. So here we focus on 30,000 healthy people with T1 brain MRI in the UK Biobank. We use as risk factors 70 subcortical and gray matter volume traits. We took GWAS summary stats for Alzheimer's as a supervisory signal, and then again trained on 80%, evaluated on 20% of individuals. These are the results. So even here, we could see we can get quite good predictions, 74% AUC. And when we looked at which traits that the model was picking up, we saw that basically the predictors capture shrinking in the midbrain, shrinking specifically in amygdala and hippocampus, and these are traits associated with Alzheimer's. We also had a third application where we aim to predict 5-year Parkinson's disease risk. So here we focus on people with accelerometry data. We consider 38 accelerometer-derived biomarkers, similar setting as before. And even here we get an AUC of 80%, and again find factors that are known to be associated with Parkinson's risk, specifically sleep duration and physical activity levels. Great, this concludes the second part. So I demonstrated that it is possible to predict future disease risk using only summary statistics from an external GWAS, without necessarily relying on longitudinal data. And I use a new method for doing this within the Mendelian randomization framework that we call Primer. And we validated these in simulations and across 3 very different settings. So we could predict type 2 diabetes risk from blood traits, Alzheimer's risk from brain structure, and Parkinson's risk from accelerometry data. What's next? I think one of the potential advantages of using a causal inference framework to learn disease risk predictors is hopefully that you learn predictors that generalize better across cohorts. We haven't shown this that, but that's something that we are doing now. And then, of course, the initial ambition that I told you about is that we want to use this framework to ultimately do discovery from imaging embeddings. And that's also something that we have in the making. With that, I'd like to thank my group, collaborators, funding agencies, and a special thank to Shubham, who drove the Histology project, Luba, who drove the Retinal project, and Daniel, who drove the Causal Inference project. Thank you very much for your attention. Look forward to questions.